So good morning once again, and welcome back. This is the course in Beginning of Life Bioethical Issues within the Masters of Bioethics at St. Thomas University. Beginning as always with a little prayer in the name of the Father, the Son, and the Holy Spirit. O Lord, send forth your Spirit, and we shall be recreated. You shall renew the face of the earth. Amen. Father, Son, Holy Spirit. Okay, let's get some blue lights here. Is that good lighting for you? You can see the screen better. And I'm going to minimize the tile so it doesn't show on the video. Okay, so we're still at the beginning of the course, at the beginning of life issues. And as always in bioethics, we are establishing first the biological facts, and then we'll do our ethical analysis based on that. So before going further, any questions or comments from previous lecture? The previous lecture, mostly I looked at asexual reproduction. So a very brief recap is we have two modes of reproduction in nature that we have observed so far throughout the world. And these are universal observations, meaning that whatever species we look at, any organism that is living today and for centuries and millennia going back, we see that all organisms fall into one or sometimes both of these categories, asexual or sexual reproduction. And there are advantages and disadvantages to each with regards to the progeny and the energetics that are involved, of course. You know that reproduction is one of the two characteristics of being alive, organically speaking, right? And the other characteristic is metabolizing. In other words, having body function, metabolism. So those are the two essential characteristics of a living organism. That the organism is metabolizing, that there are biochemical reactions going on are keeping that organism alive. And the second characteristic is that these organisms, these individuals within any species are capable of generating offspring, a next generation of the same species. Okay? So we take all this for granted, and yet the mechanisms that are involved for allowing this to happen are just short of formidable, because the layers of complexity can really be bewildering. We know that the molecule of inheritance is DNA, right? And we say it kind of fast, 3 little letters. And what is behind the whole idea and reality of DNA, again, to me, is totally mind-blowing. For example, we look at chromatin, and we talked about chromatin last time, right? How many molecules in chromatin? Say we have a cell from the skin here, and we're looking at the nucleus of a single cell, right? If that cell is not undergoing mitosis, then the cell is in what phase, what stage? In between reproductions, in between mitosis, the cell is in interphase, right? Interphase. And what is the cell doing in interphase? Metabolizing, being alive, doing its thing, you know, anabolism, catabolism, all the biochemical functions, all right? So again, think functionally, and with our mind's eye, we can get as tiny as we want. So get into a cell and get into the nucleus of a cell, and what are we gonna see around? We're gonna realize that we're inside a bowl of what? What looks like spaghetti. Spaghetti. And then let's say we get out of the nucleus and magically we reach in and we find like the end of that spaghetti, right? And we start pulling on it. How many spaghettis are we gonna get from a cell in interphase? A single spaghetti, a single strand, okay? A single spaghettum, because spaghetti is the plural actually in Latin. So we get a single spaghettum, and if we were to stretch it out in front of us, hypothetically, without unwinding double helix, keeping the double helix intact, but unwinding the supercoiling, all right? And just take it at the first level of coiling is what? The first twist is the twisted ladder, all right? So that maintains integrity of that molecule. So we leave the twisted ladder intact, and we just unwind the rest of the super winding. How long is that going to be? 6 feet, 2 meters, right? Very significant. And how many molecules am I holding? One molecule. One molecule. It's one spaghettum. Okay? And what do we call that structure in the nucleus? The DNA that is not reproducing but metabolizing? Chromatin. Chromatin, right? It's the complex between, chromatin is actually the complex between DNA and the histones to form the nucleosomes, beads on a string and all that, right? Functionally. But what I want to point out is that this one molecule that is 6 feet long, I mean, how many molecules do you know that are 6 feet long? Not even proteins, right? Which are polymers. It's amazing. One single, so how many atoms do you think that molecule has? Molecules are made out of atoms, right? The atomic theory. Okay, so what is keeping, let's break it down again with our mind's eye. We're tinkering around with this molecule in front of us, and we can get in and out of it and through it at the atomic level. We realize that the molecule is a twisted ladder, it's a double helix, so it's got a rail, 2 rails that are parallel, anti-parallel, technically, and so forth. And then we see the steps in between, right? The rungs or the steps of the ladder. What are those called? Those are the nucleotides or base pairs, and we notice that A pairs with T and C with G, etc., right? Base pairs. Okay. So some are single rings, some are double rings, but how many atoms in each one of these bases? Dozens of atoms, right? Single ring, double rings of carbon and nitrogen and oxygen and hydrogen and so forth. They're organic molecules. All right, so each base has dozens of atoms. So a base pair has dozens times 2. And how many base pairs do we have in the human genome? 3 times 10 to the 9. To the 9. 9 billion, approximately. 9 billion base pairs. That was a big human genome project. Any of you heard of the Human Genome Project? That happened, it started in the 90s. It was exciting because I was going to graduate school at that time. I was thinking about genetics. It happened in the 90s, and it was supposed to be a 10-year project to 2000 to map out the entire human genome. In other words, the 3 billion base pairs that makes up the human genome, and put it online, pump it out for free. Imagine the exorbitant amount of research can be done, medical and non-medical, if we have the human genome deciphered, right? So that was a very ambitious project. They set themselves out for 10 years. Ever heard the name Francis Collins and Craig Venter? Those were the big guys back then. Francis is still, I think, running NIH today, Collins. At any rate, when they get to the year 2000, the big millennium, they were about 99.99% with the sequencing, but they were missing still a fraction of a percentage. Now, when you take a fraction of a percentage of 3 billion, that could still be a few million base pairs. And it was not just this blank sequence in the middle, it was dispersed all over, so there were gaps. There were gaps, all right? And they actually took 1 extra year and pushed it through. And at that point, I was in graduate school from 2001 to 2005. Took about a year and a half, I think, more or less. And they had sequencers, DNA sequencers, in many labs throughout the whole world. Japan, Israel, the US, of course, the UK, all over Europe, India, all of the major developed countries in the world that had sequencers, DNA sequencers, they all got into the game. And finally, we're able to crunch out that 0.00001% that was missing to fill in the gaps. All right, and so now we have the human genome sequence, the 3 billion base pairs. You multiply 3 billion times dozens, so you're in the trillions of what? Atoms. In what? In one molecule of DNA, in one chromosome, in every single nucleus of our body. Trillions of atoms making up a single molecule. It's amazing. All through the process of evolution, you know, give it enough time and the right conditions, and this can happen and has happened, and we are the beneficiaries of it. A little anecdote from that time. So these 3 billion base pairs, the human genome represents, and how many humans are we in the world today, approximately? You know the number? I'm throwing big numbers at you this morning for some reason, I don't know why. We used to be about 7 billion, now we're getting close to 8 billion people. So again, a million is 10 to the, a million with an M is 10 to the 6. A billion is 10 to the 9. Trillion, 10 to the 12. Those numbers are so big that they would not even agree on the name of it. So our American billion, right, what we call a billion in Europe, it's 1,000 million. And for them, really a billion is only a million. So for Europe, the word billion is 10 to the 12. What we call a trillion. So we can't, those numbers are so large, honestly, we can't even agree on what to call them. You know, so you gotta be careful. We're talking about an American billion or a European billion and so forth as you go up to trillion, quadrillion, quintillion, right? Anyway, yeah, so we're 8 billion people. Getting there, all with our DNA, et cetera, all living, metabolizing. How many people, because this was not known at first, right? How many people were sequenced, were sampled for the Human Genome Project? You know, do you think they're gonna do, how many people they're gonna do? 1,000, 1 million, 1 billion? In other words, what is the sample size to represent the human population? For the human genome, you know, which is pretty much basic research at the core, because this human genome now is going to become the standard for doing all of the medical and non-medical research at the genetic level, right? So what kind of sample do you think would be representative of 9 billion, of a population of, well, we take about, let's say that at that time, maybe we were 6 billion in the year 2000, you know, in the 90s. Let's say we were 2 billion, 6 billion people. What would be a representative sample? That's a statistical question, right? What's a representative sample? If I go out, let's say that we have all these Australian pine, all these slash pines here, you know, we have about 300 on campus. So that's the whole population, right? What is a representative sample if I want to do some research on the slash pine? If I pick just one, it could be an adult or it could be a juvenile or it could be one of the seedlings. We got 3,000 seedlings just as this little sector. Is that one going to be representative? Maybe it's a weak one, maybe it's a sickly one, maybe it's a super vigorous one, you know, so we need more than one, right? A percentage is a good idea. 5%, 10%, 50%, 80%. You don't want to sample the whole population, it's impossible. So you have to establish 5%, you know, give or take. So what's 5% of 6 billion? Well, 10% would be 1 0 off, right? So it would be 600 million. Half of that would be 300 million. Easy, quick way. So 300 million people were sampled for the Human Genome Project. 1%, that's what, 1 million people? 0.5%, you're gonna drop your jaw. It was not known at first, but eventually they published it. 100 people, 100. So what's 100 out of 6 billion? You know, it's a 0.0000 what? And that is a sample that represented the whole human population and the whole human genome. So there could be variants, right, in that human genome. And that's when we get into population genetics and statistics. There's always a margin of error. Okay, it's going to be a margin of error. And we talk about type 1 error, type 2 errors, but it's all worked out and it's part of the statistics that is done. But anyway, that's the Human Genome Project, and many of those who were sampled were actually the researchers. So they had readily available blood samples or whatever, and tissue samples that they needed from themselves. They could self-consent and so forth, and that was it. So they got their genome sequenced. It was part of a little ego trip. They got their genome sequenced and pumped into the, into the database for the whole world. But anyway, so this DNA molecule is, I mean, it's just short of miraculous, how it works, how stable it is. And that's really all we get from our parents. All we get is these two tiny microscopic cells, and the action of those cells is the nucleus, is the DNA in that nucleus. And it's even haploid. We don't even get a duplicate, yeah, from each parent. And that's why genetic defects are passed on if we happen to get that defective side of the pair. Mm-hmm. And so all this to point out that sex cells are delicate, they're genetically very delicate, all right? And that's why the reproductive process is a delicate process, and it's a fast process, and it has layers of redundancy in it. Part of it is the overabundance of progeny. So there's selection. I don't know if you remember from last semester, pre-zygotic selection and post-zygotic selection, right? Because of the overabundance, simply because there could be defects, and there are defects that are passed on. That's the big risk of crunching all of the genes, all of the genetic information of every individual down to a molecule, you know, and passing it on. There's going to be high risk. And the way to circumvent the high risk is to make an exorbitant amount of copies and then let nature take care of itself, meaning natural selection. The most vigorous are the ones who are going to survive, the survival of the fittest, right? But that fitness is not just muscle fitness from the gym. What kind of fitness are we talking about? Genetic fitness, genetic fitness. In other words, the strong genes, the dominant genes, the genes that are more resistant to virus, bacteria, infections, and so forth. And yes, some of those genes are going to code also for better muscle and bone structure, but only part of those, especially for animals that are subject to predation. They need to run faster, etc. You know? But yeah, so a lot of it is about genetics. A lot. And it's a fascinating field, and we're just getting started because technology is becoming so interesting now that we can get down to the molecular and to the atomic level. Now with this CRISPR, you've heard about CRISPR, CRISPR-Cas, which long acronym. Basically, now in genetics, we have the capacity to take any DNA sequence from one species and insert it into a specific region of any other DNA sequence of any other species. From bacteria to the largest mammal to the largest whale that you can think of. And it's a very cheap technique, and we can do it up in the lab with a combination of the regulatory proteins that are related with the regulatory genes at the very beginning, at the zygote and more or less stage of development. Right? We're going to get into that today. All right, so all this to say that DNA is an amazing molecule, just really, really amazing how the inheritance is passed on in any species that we have looked at so far. So that must have evolved early on, you know, like the histones, and stayed because it worked. And nature has these principles, if you will. If it works, don't fix it. It just keeps doing the same because it doesn't get selected out, basically. It doesn't get selected out, it keeps working. So as long as it works, boom, 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 it's very pragmatical that way. All right, so asexual reproduction, yes, these animals are, and plants and bacteria and organisms are capable of generating the next generation, but they are essentially variants of clones, right? Because it's the one individual splitting up, duplicating, et cetera, and therefore there's no genetic mix. And so whatever errors, whatever mutations the parent organism had, those are on average passed on to the next generation. All right, so that could, the downside is that it could weaken that species over time, as long as the, so what do you think is the advantage of asexual reproduction, in the sense that it doesn't involve the production of gametes, right? And therefore that energy, those ATPs, are saved because they don't have to go through gametogenesis. They don't even need sexual organs for the matter, because there's no sexual reproduction going on. So all that is saved, right? And as long as the environmental factors don't change drastically, asexual reproduction is fine. But what happens when there is an environmental change, like a climatological change? Selection is going to start hammering down on what? On the variants. And some variants, remember the typical variation, right? In nature is a bell-shaped curve, but not necessarily. Most approximate the average, but then there are variants. There are some that are extremely short, some extremely long, et cetera, and we can make that same argument of polymorphism for each one of the traits or characteristics that we all have, that all organisms have. So when there is, for example, climate change or any kind of environmental change, natural selection starts hammering down, because now the conditions are more adverse. Colder winters, warmer summers. So whatever fox had more fur is going to, on average, going to survive better the colder winter, you see? And the fox that had just the average fur is going to get hit harder by that longer winter, or by that more gruesome winter. So the shorter fur or the less fur foxes are going to start dying off, and the thicker fur foxes are going to survive. You can also make the argument for adipose tissue, for fat, the fatter animals, because fat essentially is an insulator, right? In the wintertime, that's an advantage. It's also stored energy. So you see, again, thinking functionally, with clones, you have very little to no variation. Those are all essentially identical to each other, right? So the advantage of sexual reproduction is precisely the variation that will come on the progeny, because you're having a mix. Think of either pure orange juice or pure apple juice, or a mix between orange and apple. So we have 3 glasses. One glass has pure orange juice. The other glass has pure apple juice. And the middle glass has 50-50. If we drink the apple juice, are we going to taste the orange? 0. We drink the orange juice, there's no tasting the apple. But if we drink the middle juice, we can taste both. You follow? That's the mix. That's the mix. So that's the advantage of sexual reproduction. The payoff of investing, which actually costs about twice as much energy, twice as many ATPs to produce the gametes, the payoff is in the variation of the next generation. Okay, so that's basically the big picture of the lecture today. And that's how sexual reproduction came about. It's interesting. I'm not going to get into the evolution of sexual reproduction, but it's very interesting how it did come about. Essentially by chance, from some chance mutation that worked and had that selective advantage. So the organisms that began reproducing sexually survived with some kind of drastic climatological change, and it stuck to this day. All right, so we're going to look at sexual reproduction in some level of detail. Then we're going to look at embryonic development, especially the early stage.
I have a question.
And yeah, give me a minute. And then probably the ontological status, we may not get to it today, but we can pick it up at the beginning of next lecture, okay? Because it's just a gradual development here. Yes, Chris, go ahead.
George.
Oh, I'm sorry, George. Got you. Your voices sound similar.
Yeah, I was wondering why the identical twins was in the asexual reproduction. That part I'm a little confused since, well, you know, the babies don't come from asexual reproduction. So why was it in the asexual category?
Right. Okay, good question, good thinking. So we're talking about identical twins, and scientifically we call them monozygotic twins, right? Monozygotic, that word.
Right.
So that word is informative actually, because what's the other kind of twin? Dizygotic, right? And what does the prefix mean? The prefix di, di, this is the added value of the program, a little Latin. Di means two, right? So dizygotic means two zygotes. How do we get two zygotes? Zygote is the product of fertilization. What gets fertilized? The egg by the sperm. It's basic, right? Okay, so we get a zygote from a fertilized egg, and we get two zygotes from what? From two fertilized eggs, right? So those are different individuals, and that's why they can be boy, girl, 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 boy, boy. It doesn't matter because they're coming from two sperm, and the sperm is the one that determines the sex. All right, and by the way, it's not only in humans. In twinning happens in mammals, it happens in many species. But so what does monozygotic mean? The prefix mono, in this case, is one or single. So we got a single zygote, right? How is that zygote coming about? Nope. Well, let's take that case. We have one egg and two sperm hit it simultaneously, and they both pour in their nuclei. How many chromosomes are we going to have in that scenario? Let's stick with the human. The egg has how many chromosomes? 23. Each sperm has 23. So, you know, 23 plus 23 makes 46, which is the diploid number, all right? But if we have two sperm simultaneously fertilized in the one egg, we have 23 chromosomes from the egg, 23 chromosomes from one sperm, that's 46, and 23 chromosomes from the other sperm, 46 plus 23, quickly, 69, right? Does that code for a normal embryo? 69? No, sounds very abnormal, right? Because 46 is normal. In fact, when you think about it, again with the mind's eye, what's happening there is that embryo, that zygote, is grossly abnormal, genetically speaking. And it actually is coding for 1.5 stuff. So if that were to express, if all those genes were to express, that embryo would have 1.5 heads, 3 arms, 3 legs, 1.5 kidneys, 1.5 hearts, et cetera, right? Because all the genetic information is in that 6-foot molecule to generate the entire individual of whatever species. So grossly abnormal. So what's your thought? What do you think is going to happen as that zygote begins? It's actually a triploid zygote, right? It begins to develop very early on. It's going to run into massive problems of expression, and it's going to die, and it's going to be discarded. And that is called a spontaneous abortion. All right, and most spontaneous abortions happen in the first couple of weeks of pregnancy. The mom doesn't even know that she was pregnant. Maybe she thinks she's just late in her period, but she's having actually a natural loss. So gross abnormalities are taken care of by nature, natural selection early on, all right? And obviously there's no guilt in that because she didn't procure it, all right? They were just genetic defects. I'll talk more in detail about that when we get to the lecture on abortion in a couple of weeks. But for now, we go back to the monozygotic twin, which was a single sperm fertilizing a single egg. And then after fertilization, what happened to that zygote? Exactly, divides in two. It could be at the first cellular division, at the second cellular division. You notice when the zygote is dividing, it essentially goes into mitosis, right? And it's always a duplicate number, so it's always an even number. And so there's a window there that I mentioned, at least for the human, up to 2 weeks of that little embryo developing. That little embryo can split into two, and we get two identical twins. So back to your question, Jorge. The twinning is happening before fertilization or after fertilization? After, right? After, right? Exactly. So if the twinning is happening after fertilization, then sexual reproduction has already occurred. It's done, over. When did sexual reproduction happen for those twins or for that couple? When there was fertilization. When there was fertilization. Right? Does it make sense, Jorge? So the sexual reproduction, reproduction is interesting. I'm getting an echo here.
But yes, I got it. Thank you.
Okay, so really we go after sexual reproduction has already happened, then twinning may or may not happen, but the twinning itself is asexual. Okay, all right, super. So let's move forward. Again, some basic stuff. I mean, this is going back to high school biology, but just in case someone missed that lecture back then, sexual reproduction is involved in the process of fertilization, okay? And keep in mind, fertilization, it can be internal or external fertilization. Now, we humans and mammals and many animals have internal fertilization, all right? But external fertilization also happens in many other animals. The female essentially lays out the eggs in the environment, typically water, or it could be land in a moist area, and then the male lays out the sperm, and there's external fertilization. So the fact that there is sexual reproduction and fertilization doesn't mean necessarily that there is copulation, that there's internal fertilization, okay? We're talking here at the cellular level. All right, so whatever type of fertilization, whether it's internal or external, to the female, it involves fusion of gametes, all right? And this is the only case where you have two cells coming together and surviving, in fact, flourishing into the next generation. Because the typical process in nature is for the cell to split, right? From one cell to, like mitosis, but from 2-1, it's the opposite, it's the reverse of mitosis. Right? So it's very interesting that sexual reproduction actually involves the coming together, the fusion of two distinct cells, which we call gametes, into one functional cell. So we can say that at fertilization, the diploid number, 2n, is restored. So as far as genetics is concerned, the purpose of sexual reproduction is to restore diploidy. We talk about restoring diploidy. We have these little phrases, right? Contractions. So fertilization restores diploidy. So the gametes are simply the sex cells, which are sperm and ova. And they are produced by a process that is called meiosis. Okay? So meiosis sounds like mitosis, but it's like a double mitosis, but with a twist. all right, with a little twist. So it's a double mitosis, but with a twist. That's meiosis, because we need to reduce the number of chromosomes from 2n to 1n. Remember, the product of meiosis is gametes, which are haploid, which are N, right? Unpaired chromosomes. So it's a very mechanical thing, and a lot of people get lost in meiosis, but really when you think about it, you just have to think like a double mitosis with a little twist, and I'll tell you what that is. So let's continue here. To understand meiosis, then we need to do a very basic review of mitosis, which is just cellular splitting. Here's chromatin. Then the cell is in interphase, and of course is diploid. When a cell starts the reproduction cycle, which is this one, all right? The four phases of the reproductive cycle that you put in your summaries and so forth. This very first phase here, prophase, is the crucial one, because in order for, okay, let's start from the end. At the end of mitosis, from one cell, how many cells do we get? 2 is a split from 1 to 2. However, at the level of DNA, the mother cell is diploid, right? And the 2 daughter cells, each one is also diploid. Okay? So was there any magic here that one of the cells got the extra chromosomes just out of the blue sky or something? No, there's no magic involved. There's mechanism, all right? And so at some point there has to be a doubling of the chromosomes so that when we get to the last stage of mitosis, that double number of chromosomes gets distributed evenly into the 2 daughter cells. And that's how the daughter cells end up with the original number of chromosomes, which is diploid for whatever species. So N just stands for number, and it's going to be a particular number for each species. N for us, for humans, is what? 23. 23, right? 2N is diploid, but just N by itself, just the N. Because N, again, this letter is universal. What is N in the chimpanzee, for example, in pan-troglodytes? N is actually 24. They have 1 extra chromosome from us. So what is 2N? What is a diploid for the chimpanzee? 48, 24 times 2, okay? All right, and so on and so forth for species. You've heard of nematodes. Some nematodes, their N is actually 100. They have 100 different chromosomes. And so 2N is 200. They have 200 chromosomes, not genes, chromosomes. So they have way more, they have 10-fold more chromosomes than we do. And our nematodes, our roundworms, more smart than we are? No, they're certainly, they're not in the higher animals, okay? They're not even vertebrates, they're invertebrates. They don't have limbs, they don't have even a full brain, they have ganglia. So the number of chromosomes, the number N, doesn't necessarily mean a higher species, all right? Generally, but not necessarily. The fly, Drosophila, has N is 4, okay? All right, so this is the duplication that happens here. From interphase to prophase, there is the duplication. Now again, in science, we don't want people to understand what we're talking about, so we make a sophistic name for it, which is replication. Replication, all right? DNA replication, remember DNA duplication, DNA replication, this is what's happening, right? So the number of chromosomes are doubled, just doubled, which means that temporarily this prophase cell, what is going to be N? Is going to be 4N, temporarily 4N, because each chromosome has duplicated. So each pair has duplicated. So we have 4N. So temporarily we have 96 individual chromosomes there. 23 times 2 times 2. So temporarily in the prophase, we have 4n. All right? And so I'm just putting numbers to them to be identified. So 1A and 1B is one pair, and 2A and 2B is the other pair, but you can see that they have duplicated themselves already. They're being held together by this little white dot, which is called the centromere. And then they line up, the pairs line up at the equator of the cell. And then and that's at metaphase, meta meaning middle. So that's the lining up at the equator of the cell, all of the chromosome pairs, the duplicates, all right? And then in anaphase, they split, the centromere gives way basically, and each chromosome individually goes to the opposite pole to form the new nucleus of the two daughter cells. So here's a little bit more detail. This is the cell in interphase that is about to duplicate, is about to undergo mitosis. And so this is DNA replication has already happened. This was the original chromosome of each one of the 23 pairs, right? This is before the split. This is after the split, after the replication, right? So replication happens here, and those legs or those pairs are held together by that centromere that I talked about. Each one of these legs is what is called a chromatid. You may have heard that name, chromatid. But essentially you have, at this stage, you have really 92 chromatids, 92 chromatids. Let's go a little further, the diagram. So that process is called DNA replication. There's a third name for it. So we got DNA duplication, DNA replication. It's also known as DNA synthesis, because synthesis is making something, just like protein synthesis is making protein. Don't get confused, little. Anyway, synthesis is the same as duplication or replication for the DNA, right? Here are the chromatids, the consistent chromatids, because they're identical to each other. Here is the whole cycle put in place. It starts at G1 here, and the G stands for gap. Then here is this synthesis part, the DNA synthesis, replication. You can see you got single chromosomes here, you got the duplicates, and then the cell goes into what is called G2, gap 2, and this is like a double check, it's a gateway to make sure that DNA synthesis has happened indeed. And if DNA synthesis has happened, if replication has happened, and we got sister chromatids in the nucleus, and in other words, we got the 92 individual chromosomes, then the cell finally goes into mitosis. And the end of mitosis, we have the two daughter cells, each with the diploid number, okay? And those will be identical to the mother cell. And that's DNA, that's mitosis in some level of detail, put within what is known as the cell cycle. Then each daughter cell at the proper time will enter its own cycle again and again and again, and keep splitting and duplicating. And that's how from one cell we get trillions of cells over a period of time. This happens a lot all the time as we're alive, but it happens very fast, especially in the embryonic development, because we have to go from one cell that is microscopic, and 9 months later, we have a baby who's about 1 foot long or 1.5 feet and has already trillions of cells. And all that has to be done in 9 months, okay? So super fast activity. That's one of the reasons why the heartbeat of a baby, of an unborn baby, you know, what is the heartbeat? What is the normal heartbeat of a human for us right now? heartbeat is the same as the pulse rate, right? So we have an average pulse rate, 60, 70, 80 range. If we have a low pulse rate, like below 60, 50s, for the young people, that is an athletic predisposition because the resting heartbeat is low, right? So the heart is unstressed. And when we do exercise, we speed up the heart rate, right? Let's say we speed up to 50%. Let's say we have a pulse of 60, and we speed up to 50% more. Now we're at 90 beats per minute, right? The pulse, okay? So the heart is just taxed a little bit because really the normal is around 80, whereas a person who has an 80 heartbeat and they speed up 50%, 80 plus 40, now their heartbeat is at 120. So you compare 90 beats versus 120 for every minute of that heart. Which heart is more taxed? The 120 heart, right? In other words, the 80 pulse. And that's why a low pulse for the youth is an athletic predisposition, whether they're going through athletics or not. They can be a couch potato all their life, but they have an athletic predisposition just by having a low pulse, which is typically a genetic thing. It's an inheritance, all right? So people who have low pulse, generally, they have an athletic predisposition because they can speed up the heart into what would be for that heart a normal rate, as opposed to another person who is now stressing their heart, okay? As an old age, that advantage that was good for the youth is now a disadvantage for the elderly. I'm talking about 60 years of age or over, because what happens? That heart is beating more slowly and slower and slower and slower. And you're going to go from 60 to 50 and from 50 to 45. And now the person is becoming lethargic because they're not getting enough circulation, especially to the brain, which is the higher floor, it's the penthouse of the building. All right, so it takes more effort to get that elevator up there, to get the blood up there. And that's where we need most of the blood, 60% of the circulation through the brain. And so we get less oxygen. And so we get tired, we slow down, we can have moments of blank out. And so the elderly who had a low heartbeat when they were young and was an athletic predisposition and a fitness advantage now becomes a disadvantage. And sooner or later, we need a pacemaker prematurely, like maybe at 70 years of age instead of 90 years of age for the pacemaker, okay? So if you have a low heartbeat, take advantage while you're young. I know that because my heart is about 50. All right, here we go. Now, let's compare mitosis to meiosis. Here is mitosis, a simple split, one single split. We start with 46 chromosomes. We end up with 46 chromosomes in each daughter cell. So diploidy is maintained because of the duplication that happened early on in the prophase of the thing. Actually, in the synthesis phase of interphase, just before prophase. All right, fine, you got all that. Now, meiosis. We start with the same diploid cell, but there's a double split. So we end up with 4 cells instead of 2 cells, right? That's why I say it's a double mitosis. This would be the first mitosis, then a second mitosis, integral part of the whole process of meiosis. But each daughter cell, each one of the 4 daughter cells is only going to have 23 chromosomes. And that makes them haploid. So there's no pairs in the gametes. All right, so the end result of meiosis is these sex cells that we call gametes, egg or sperm, and that's how we have this process that is called chromatic reduction. So again, you know, because we want to be cryptic and don't want people to understand us, this is what is called chromatic reduction, because we have reduced the number of chromosomes to the single number, to the haploid number. And the pairing has been redistributed into the 4 daughter cells. All right? mathematically is the same, because if we were to do a second split here, hypothetical, if we were to do a, excuse me, if we were to do a meiosis split on these daughter cells, we would also end up with 4 daughter cells with 26 chromosomes each, right? 23. You follow? Okay. So I said it's a double mitosis. Meiosis is a double mitosis, but it's got the twist. And the twist is really to get this haploid number, to get them unpaired, to get the chromosomes to unpair. You with me so far on the arithmetic? All right. The twist is over here in the first mitosis, okay, early on. The second split is essentially a straight up mitosis, but the twist is on this first split here. Let's look at it a little bit in more detail. All right. So, see, here's the first split, meiosis 1. The second split is called meiosis 2, okay? And I'm telling you that the twist, the tricky part is here. First, interphase, replication from each chromosome duplicates, generates sister chromatids. So we got the sister chromatids being held by the centromere. And now we're going to pump in here on the diagram. Now, you notice that in the diagram here, the chromosome pairs stay together. Think about what I'm saying. Chromosome pairs stay together. So chromosome pair 1, you know, we got 1A and 1B. They have already replicated, so we got sister chromatids, but those chromosome pairs stay together. So really, when we count the number of chromatids, each chromosome has 2 chromatids, right? So when we count the number of chromatids in the chromosome pairs, we have how many chromatids? 2 plus 2, 4. 4 chromatids altogether. And that is called a tetrad. So tetra for 4, all right? That's a tetrad. The tetrad complex is 4 sister chromatids that come from 2 chromosome pairs, all right? And those tetrads line up together at the equator in meiosis 1, okay? Excuse me. Whereas, I'm going to change the diagram here for a moment, whereas in good old mitosis, you notice that the chromosomes line up singly. Yes, we have sister chromatids, but the chromosomes are lining up at the equator singly in mitosis, not in pairs. There are sister chromatids that are being held together by the centromere, but the chromosomes themselves are lining up singly in mitosis at the equator in metaphase. Whereas in meiosis, they line up, the chromosomes line up in pairs. So 1A and 1B stay together, 2A and 2B stay together, 3A and 3B stay together for all 23 pairs. All right, so it's a very ordered process. They really stay together, those pairs. Of course, each one of the pairs has already undergone replication. So we got the chromatids, the sister chromatids, and that's why we talk about tetrads. Tetrad is important also for another thing called recombination, which we'll look at in a minute. All right, so they line up in pairs, the chromosomes line up in pairs at the equator, and then when they split, the pairs split to opposite poles. The pairs split to opposite poles. But what happens is that the sister chromatids stay together. The sister chromatids for each chromosome stay together. So some splitting has to happen to go to opposite poles. But the splitting is the splitting of the pairs, not the splitting of the sister chromatids. So essentially the centromere stays intact. Compare this diagram Again, back to mitosis. I'm going to go back for a moment. You see what splits here is the sister chromatids. What splits is the sister chromatids. So what gives way here is the centromere. And the sister chromatids go to opposite poles, right? Whereas in meiosis, because the chromosomes lined up in pairs, what split is the pairs split. But the chromatids stay intact with their centromere. So in effect, after meiosis 1, at the end of meiosis 1, we do end up with 2 daughter cells, but each daughter cell has 23 chromosomes, but with the sister chromatids already replicated, right? So at the beginning of meiosis 2, we have 2 daughter cells because the cell has undergone 1 split already. And when you think about the nucleus, each nucleus of these 2 daughter cells, each nucleus only has 23 chromosomes because each nucleus inherited 1 of the pairs, right? But those chromosomes have already undergone replication, so the sister chromatids are there with their centromere holding them together. Okay, and there's no replication again. Why is there no replication again? Well, when you think about it functionally, let's go back to the cell for a moment. In the synthesis part, that replication involves a machinery getting in there and duplicating the chromosomes, right? The replication machinery. But because at this stage, we already have the chromosomes super condensed, the most condensed possible. The replication machinery cannot get in there and replicate again. So it's done. There's only 1 replication that went on originally for the original cell, the precursor of the gamete, okay? They're called germ cells, by the way. These germ cells are the ones that we have in the testes for men and in the ovaries for women, all right? The follicles. So when we get to this stage, at the end of meiosis 1, we in fact have 2 daughter cells. But the daughter cells each, in their nucleus, they have only 23 chromosomes each, and those chromosomes have already replicated. Now, each one of those daughter cells undergoes essentially mitosis, In other words, this daughter cell, let's follow 1 daughter cell, the nuclear envelope dissolves, the chromosomes line up at the equator singly because there are no pairs, right? The sister chromatids are still held by the centromere. Then at anaphase, the centromere gives way, and each one of those chromatids heads for the opposite pole. So essentially, it is a straight up mitosis that is happening in meiosis 2. And then the end result is that for each daughter cell, we end up with 2 cells. For each daughter cell, we end up with 2 cells that only has 1 set of chromosomes, namely the chromatid that went either to the North Pole or the South Pole. Okay. So each original daughter cell is going to generate 2 daughter cells because mitosis duplicates the number of cells. However, because in meiosis 2 we started with 2 daughter cells to begin with. We started with 2 daughter cells. The end result is 2 plus 2 is 4. So that's how we get 4 gametes for each germ cell, for each precursor, 4 gametes. And each one of those 4 gametes only has 23 chromosomes. One way of looking at it, it only has 1 set of chromatids, of sister chromatids. All right. Now, I've used a lot of technical language here, but the diagram is very succinct. And you wouldn't believe how many people get confused with meiosis because they fail to realize where's the twist. The twist is early on, and the twist is lining up at the equator in chromosome pairs. Whereas in mitosis, they line up at the equator, the chromosomes line up at the equators, but singly. Yes, we got sister chromatids, but those chromatids are still together. So the chromosomes line up singly in mitosis. In meiosis, they line up in pairs, so that here is the bottom line, the chromatic reduction occurs here in this step, okay? The chromatic reduction occurs here because when you think about it, this cell, this original cell is diploid, but the two daughter cells are now haploid, you see? And that's why in the second split, haploid stays haploid because each daughter cell gets one chromatid, which that chromatid is, functionally becomes the chromosome. So the chromatic reduction occurs in meiosis 1 and occurs between metaphase and anaphase of meiosis 1 when the pairs split to the opposite poles. And that's the whole thing. That's the whole thing about meiosis, okay? Now, functionally in situ, where is this happening? This is happening in the sexual organs, again, for female of whatever species, in the ovaries, for males of whatever species, in the testes or testicles, right? And that's their function. That's their, for the sexual organs, that's their function. To generate gametes from precursor cells, which we call germ cells, all right? And the germ cells are lining up either the ovary or the testicles to produce these gametes. Stop, question, comment, anything you can follow? You can follow the genetics, you can follow the arithmetic, you can follow the cellular division, all right? Let's go forward. Let's look at this original cell. Well, this is okay, so again, this is comparing the two diagrams. There's a single split here, and here's a double split. One split here, second split here for meiosis. But I want you to look at something now. Remember how I talked about the tetrad? Is that diagram large enough? Can you actually see a tetrad here? Maybe I can make it a little larger. Okay, now I need my bar. Here's the bar. Gotta be careful. Okay. So here's a precursor cell. This would be the germ cell. Either this would be a follicle in an ovary. This would be the lining of the tubules inside the testes. And I think it's large enough. You can see this is a tetrad, right? Because it's got two chromosome pairs that have already replicated. So we got how many total chromatids in each one of these tetrads? 4 chromatids, right? 4 chromatids, 2 pairs of sister chromatids. Let's say this is chromosome 1, this is chromosome 2. Well, okay, now look at this little diagram of the tetrads, and then the end result down here. Look at these. See how this chromosome is all blue, but this one has a red tip, and then this one is all red, but this one has a blue tip. Okay, that is a process that we call recombination. That's the variation. This is the advantage of sexual reproduction. One of them, one of the 2 advantages. This is one, which is the recombination. So let's go back for a moment. In our cells, in our diploid cells, we have chromosome pairs. How many chromosome pairs in this diagram? How many? 2 pairs. Pair 1 and pair 2, right? How many total number of chromosomes? 4, because 2 pairs is equal to 4 individual chromosomes, okay? So they replicate, they form the tetrads. So here is chromosome pair 1, chromosome pair 2 in tetrad formation. Then they do something that is called crossing over, or The fancy word is recombination. So you've heard of DNA recombination. This is it. Let me show you a bigger diagram for illustration. A little smaller. Okay, so here is the recombination. This is chromosome pair 1. Has this chromosome 1 replicated or not? Yes, replicated, because here's one sister chromatid, here's the other sister chromatid. This is the centromere, right? So let's say that this is chromosome 1, pair 1, 1A, 1B, whatever. They have already replicated. So we have a total of 4 chromatids. They pair up at the tetrad. This is the tetrad. And then they cross over. In other words, they exchange pieces of chromosome, literally sequence, all right? They exchange. I give you my hand and you give me your hand, and I'm gonna cut off my hand and stick it into your arm, and you're gonna cut off your hand, and I'm gonna stick it into my arm. And we exchange hands, all right? So that is done, again, at the biochemical level. You can imagine the level of sophistication of the machinery that does this, and there is a machinery that does this, all right? It's a complex of DNA molecules and so forth. And this is the crossover, the recombination machinery. The recombination machinery. At the molecular level, it involves, think of 2 train tracks that are next to each other. It involves cutting one track of the train and cutting on the other track of the other train and relinking them, and then doing the same on the other rail. And so that when the trains come, the one train will cross over to the other track, and the other train will cross over to the other track. Okay, so it's the rails of the twisted ladder that get cut off and rejoined in the sister chromatid. So this is recombination. Functionally, the blue genes go to the red chromosome, and the red genes go to the blue chromosome. Now, what are these genes? These are gene pairs. Now, the gene pairs could both be dominant genes, or both could be recessive genes, or one could be a dominant gene and the other one could be a recessive gene, all right? And the advantage happens when the recessive gene gets replaced by a dominant gene. It's the same gene that codes for, I'm gonna exaggerate, make the eyeball, okay? But it's the difference between making a very sharp eyeball or a very colorblind eyeball. Let's say the dominant, I'm just making it up, the dominant gene, eyeball gene is to make a very sharp sight, very good sight, all right? Of course, there are hundreds of genes involved in making an eyeball, but let's just reduce it to one. And the weak gene, the recessive gene, is a colorblind eyeball gene, all right? So in the recombination, the recessive gene gets replaced by the dominant gene. So a child that would have had color blindness now has sharp sight because that child happened to get the recombinant DNA. The opposite can also happen. The weaker gene, let's call, I don't know, either one, let's call the blue the weaker one. Originally, whoever gets this gene, this chromosome package, this DNA package, would have gotten a dominant red gene. Now they're going to get a recessive blue gene. So they're going to get a weaker. So it can go either way, all right? But the advantage, of course, is on the dominant, because that's going to make for, this is fitness. This is the genetic fitness I was talking about. So recombination allows for the possibility of strengthening the progeny. It's up to natural selection then to have it express or not. That recombination happens early on. Again, it has to happen here at prophase when you have the tetrads. By the time these tetrads line up at the equator in metaphase, recombination event, it's over, because now they're getting ready to go to the opposite poles. And of course, once they split and go to the opposite poles, that recombination matter have finished. Otherwise, they're gonna have a piece, the arm is gonna be torn off. Okay, so recombination is one tremendous advantage of sexual reproduction. It happens in early on in prophase 1, precisely, at the tetrad stage. And that's the big advantage. That's the big advantage where the extra energy is spent to make sexual reproduction successful, because it introduces variation. It's one of the ways that introduces variation down to the chromosome level. The other way of introducing variation is at the level of the entire package. Since we get 50% of the genes from our mother and 50% of the genes from our father. So that's the second layer of variation that we get, you see? And so there are two layers of variation, really, that we get in sexual reproduction. One is with recombination, and the other one is with the mix, the haploid mix of father and mother to restore diploidy. Okay, a lot of detail here. What I'm trying to point out really in the big picture, if we span out for a moment, is that there is an elegant mechanism for maintaining both fidelity and novelty simultaneously. Fidelity in the genome as a whole, because what I get from my parents is the human genome. I don't get a monkey genome or a giraffe genome or a butterfly genome. I get the human genome from my parents, okay? But I also get variation in the process of meiosis. That double variation, when they made their gametes, automatically in my mother's ovary and in my father's testicles, and then also in the actual individual egg that got fertilized by the individual sperm. So that double variation came there. So you notice that the elegance of sexual reproduction is that there is fidelity with regards to the species as a whole, but also variation within the species, which we don't get in asexual reproduction, all right? And that is why really sexual reproduction is so spectacular, because it introduces variation within the progeny within the same species, maintaining the species as a whole, maintaining the genome, all right? Good, so that's the advantage. And that advantage typically is no big deal. You know, who cares if I'm 5'2" or 6'2" feet tall? Until I have a lion chasing me, then the longer femur is going to be a selective advantage, definitely, right? And the pygmies are going to be eaten up, and the slangy, what are they called? There's another word for them. In the African tribes, the Aboriginal tribes, the ones that were very tall, you know, sometimes 7 feet tall, basketball players eventually, they got selected out, the little ones as opposed to the tall ones, okay? All right, so now let's move forward a little bit with a little more vocabulary. Meiosis then is formation of gametes, and the gametes are haploid, meaning that they're unpaired, but it's the full genome is there. And functionally then, because it's making gametes, the process is called gametogenesis. Genesis is a reference to origins or beginning or making, right? So gametogenesis is the formation of gametes. But of course, because the gametes are either sperm or egg, then we have spermatogenesis for generating sperm, and oogenesis for making eggs. Oogenesis, all right? Left over from the Greek, going through German, Germanic Teutonic language, oogenesis. I guess future generations will drop on O. All right. So again, the cells. Now, another twist. Eggs are expensive. Sperm are cheap. Again, cellularly speaking, in the sense that a female, whether human or not, on average, let's stick with mammals, produces 1 egg. Back to the human, since we're looking at human bioethics. On average, in every cycle, a woman will produce 1 egg. All right, but it's meiosis, so there should be really 4 eggs. We're going to look at what happens to the other 3, which are called polar bodies. Whereas for sperm, for every meiosis, the man will produce 4 sperm. Okay, and each sperm is equal to each other in morphology and so forth. So this is what happens in, why is it that women only produce on average 1 egg per cycle, whereas on average for every germ cell, we produce 4 sperm? Well, because there's an uneven distribution of cytoplasm. Again, in meiosis 1, in the egg. So here is the precursor, also known as the primary oocyte, has undergone replication already. We see sister chromatids, we see actually the tetrads. Now, at the end of meiosis 1, there is this phase that I've talked, really haven't mentioned it much. The splitting of this cell at the end of each cellular division, the splitting is called cytokinesis. Cyto because it's a reference to cytoplasm. Kinesis because kinesis is splitting, right here. The split, you see it here, cytokinesis. The splitting of the nucleus is called karyokinesis with a K, K-A-R-Y-O, karyo. That's Greek, it's a reference to nucleus. Karyo is a reference to nucleus. So karyokinesis is what I've been focusing in because of the redistribution of chromosomes. But the end stage of the splitting, the actual splitting, is a pinching effect of the cytoplasm in between. And that pinching effect is called cytokinesis. Again, kinesis meaning splitting, right? And it's the cytoplasm that's splitting. Well, it turns out that in the female reproductive cycle, that cytokinesis is uneven. It's not an even split. You can see here for the sperm precursors, there's an even split. In other words, this spermatocyte has as much cytoplasm as this other spermatocyte, all right? But in the egg, this oocyte has more, much more. In fact, this one is even not shown proportionately. If this were shown to real life size, this cytoplasm would be even smaller, just like a little dot, okay? Much, much smaller. So there's an uneven distribution of cytoplasm in egg formation. Has several advantages. First of all, the egg, at the end result, the egg itself, the ovum, is about 10 times larger than the sperm. The egg has a lot of cytoplasm compared to the sperm. But again, because there's no magic, how did that egg get so much cytoplasm? Because it essentially cannibalized the other 3 daughter cells, the cytoplasm of the other 3 daughter cells. You know, it sucked up more cytoplasm from the other 3 daughter cells, okay, to the disadvantage of the other 3 daughter cells. So what happens? What is the disadvantage? That the other 3 daughter cells ended up with very little cytoplasm, so little that they're non-functional, they're not viable, because it's essentially just the nucleus and a little cytoplasm around the nucleus. And they're all bunched up, crunched up, and they don't have a chance to get fertilized. So what do? They just stick around at the edge of the one egg that is viable, and they're called polar bodies, all right? And I'll show you actual photographs of polar bodies in a few minutes. So that's what happens basically after first mitosis. One of the cells, of the two cells, cannibalizes, essentially sucks up more of the cytoplasm, and the other cell becomes shriveled, and then it does that again on the second split, so that we have one cell ending up with most of the cytoplasm and the other three very tiny polar bodies that are just going to stick around at the edge. You know, they're not real parasites, they're just hanging on there at the edge, and they do nothing. They have their DNA, they have their full genome actually, but they're all crunched up and shut down, and they're not going to express at all. And they're not going to get fertilized either. Okay, and again, this is all regulated by regulatory proteins, which are coded by regulatory genes and so on and so forth. So you can see that the level of sophistication here only goes from one sophistication to the next to have this happen in eggs and not in sperm. Part of it is because the egg stays put inside the reproductive system, but the sperm has to travel. And so the sperm has to be motile and doesn't want to carry about too much cytoplasm because the cytoplasm gets in its way. It's like a big fat fellow trying to move through the weeds, you know, it's going to get stuck. And so a slim fellow can move through the weeds easier. And also because the cytoplasm part of it is going to go into a very specialized cell that is a motile cell that has a flagellum, has a tail. All right. So that's a difference in gametogenesis between spermatogenesis and oogenesis in the morphology of the gametes. Okay, back to a little arithmetic here, just to make sure that we're on the same page with the numbers. And for the human specifically, the number of chromosomes is 46 or 23 pairs. This is total number, all right? But this 23 pairs, I put it in quotations because it's only identical really in the female, because the 23rd pair for the female is truly XX. But for the male, the 23rd chromosome is the 23rd pair XY, okay? And that's why 50% of the sperm is going to be X sperm, and the other 50% of the sperm is going to be Y sperm. And then it's just a question of which sperm gets to fertilize the egg first, whether it's an X sperm or a Y sperm, and that's going to determine whether the baby is a boy or a girl. And on average, it's like flipping a coin, and that's why we have more or less 50% men and women in any population, any natural population. And then when you have governments or other mechanisms interfering in that and disbalancing that normal natural ratio of 50% each, big social problems occur. Like for example, China with their one-child policy. I'll talk more about it when we get to abortion, but they have social disaster by doing this one-child policy because they have favored the men. There are many more men than women now because couples naturally select out women, babies. Females are harder to raise, et cetera, et cetera. We'll get into that a few chapters away. It has big, big social implications, big, huge bioethical implications, okay? It's called genetic engineering of the population, and it's bad news. Okay, so we understand the numbers. N is equal to 23, so 2n is equal to 46. The haploid cells are called gametes, egg and sperm, and the other cells are called somatic cells. Somatic cells because they come from, they make up the soma. Soma is the body. Soma is now, I forget if it's Greek or Latin, but it means body, okay? Body cells. So the vast majority of our cells are diploid, or somatic cells, except for the gametes. Now in situ, here is the testes, here is the ovary. Let's make it a little larger so we can see the actual tissue, the germline tissue. For females, there are the follicles, and each follicle matures one at a time on average every month. Sometimes 2 follicles mature simultaneously. How many eggs are going to be released? 2 eggs. And on average, if there is copulation, there's millions of sperm swimming around. So you end up with dizygotic twins, right? Dizygotic twins, etc. Okay. In the testes, they're called the seminiferous tubals. They're literally inside the testes, all right, and they're protected. And these are tubals that go back and forth and coiling. And in the inner lining of the seminiferous tubals is the production, also the germ cells, they're equivalent to the follicle for the woman. And they produce, through meiosis, they produce the actual sperm, all right? Start with a spermatogonium, which is diploid, and then the splitting, the primary spermatocyte and the primary oocyte are also diploid. And then in the first split, the first meiotic split, I explained that's the chromatic reduction, all right? So we go from 46XY to 23X or 23Y. In the woman, here is, here is the first meiotic division, and it's not showing, the polar bodies are not shown here. It's deceiving because this actually looks like the nucleus. But this is the chromatic reduction from 46XX to 23X. Now, let me explain this for a moment because this is, again, this is genetic notation, 23 is the number, right? Or 46, 23 is the haploid number. What does this X represent? First of all, let's go back here. This does not mean 46 chromosomes plus XY, because that would actually make it 48 chromosomes, okay? It just means that it's showing that this is a male. So the comma and then the letters it's just showing the sex chromosomes. That's all it's doing, it's the notation, but it's still 46 chromosomes. So one of these 46 chromosomes is the XY pair, or one pair. Whereas in the female, they only have X chromosomes. The sex chromosome is only X, and that's why that pair can only be XX. And that's why the woman can only produce X, chromosome, X eggs, because the woman herself doesn't have Y chromosomes, whereas we have Y chromosome. So the 23rd pair, the sex pair, is XY. Therefore, at the first split, one of the daughter cells ends up with the X X chromosome, and the other daughter cell ends up with the Y chromosome. Then they undergo the second split. The X cell splits, X and X, and the Y cell splits again, Y and Y. And that's why at the end, these four sperm here represent the millions of sperm that we produce from millions of cells that are germ cells that are reproducing. But the point is the ratio, the 50%. So 50% of our sperm is X, and the other 50% is Y. Before moving into the last part of this first section, we're getting close to a break soon, I promise. I want to talk about the numbers functionally, all right? So again, think functionally, let's go to functionality, at least for the human. In every cycle, a woman on average will produce how many eggs? 1. And how many polar bodies? 3 that are non-functional, attached on the edge of the egg. So 1 functional egg, 1 functional gamete on average. And that cycle typically is a 28-day cycle. It's 4 weeks. It's related to the moon actually, and the gravity of the moon and so forth. But it's on average, it's a 28-day cycle, once a month. All right. For men, once the testes become functional in the teenage years, during puberty, just like the woman, when she ovulates her first egg, that's called menarche, right? The first egg ovulation. For the men, functionally in the testicles. In the seminiferous tubals, we actually have literally thousands, maybe hundreds of thousands of germ cells in spermatogonia, that when they become functional, in other words, when we reach puberty and begin to produce semen, there are thousands and hundreds of thousands. And each one of those cells is going to produce 4 sperm, all right? So we really end up with millions of sperm in each semen ejaculate. So I'm talking clinically. Think about this. Hypothetically, how many sperm are necessary to fertilize one egg? 1:1 ratio, right? Because we don't want abnormalities. So one sperm is sufficient to fertilize one egg. However, clinically, in order for a human ejaculate clinically to be considered potent, in other words, capable of fertilizing, do you know how many sperm are necessary? That's what they do. That's what they do, the sperm count. Some men have low sperm count and they have difficulty fertilizing, okay? Functionally, think about this, functionally, what do you think is the number? Hundreds, thousands, hundreds of thousands, millions of sperm in each ejaculate? Millions. In fact, 100 million on average. 100 million sperm is considered potent, clinically speaking, you know, for that ejaculate to be capable of fertilizing the one egg. So talk about competition. The race is like, you know, the marathon, the New York Marathon may have, what, 1,000, 10,000 people, 40,000 people? The LA Marathon, okay? Well, one is going to be the winner. The next guy or lady, whoever, will be a second away or a mile away or 3 hours away, but the first one is going to be the winner. The same here, only you got 100 million sperm taking off at the gate and swimming like gangbusters, but they have to get all the way to fertilize the egg, all the way at the end of the fallopian tube. So proportionally, being that the sperm is a macroscopic cell, proportionally, that is equivalent to running hundreds and hundreds and hundreds of miles to get to the goal, proportionally. Even though anatomically, it's only a few inches. It's maybe 5 inches, 5 to 10, it's less than a foot, much less than a foot, all right? Okay, so I'll leave it there. We're gonna get into that in the second half, but let's stop here. Questions, comments, anything? Everybody's been following very well. Thank you very much. Okay, let me pause and let's go thaw out for a little bit because it's freezing in here. Okay, folks, welcome back. So we left off with gametogenesis. Essentially, we can summarize the past 2 hours in this one word, gametogenesis, which is the formation of gametes, egg or sperm. And now we're ready for the actual sexual reproduction itself, the process of the fusion of egg and sperm. Okay, so that fusion, excuse me, that fusion of egg and sperm will generate a new cell that is called the zygote. The zygote, and the zygote comes from Greek zygon, which means yoked or linked. All right. And it's very symbolic because if you think about it, these Greeks back 2,000 years ago, even before then, really didn't have a clue of sexual reproduction at the cellular level because the cell theory was not around yet. They didn't have microscopes or anything like that. But they intuit that there was some combination of male and female into this new individual. And actually the linking or the yoking is at 3 levels. I'll talk about that in the second half now. But basically what I want to establish, again, just reiterate that the zygote will have 50% of the genetic material from the mother and the other 50% from the father, okay? Because of the restoring diploidy, half of the chromosomes come from the mother and the other half come from the father, from the 2 cells that fuse together. All right, I want to show you a little video that I found some time back on the internet. And this is a couple, it's a husband and wife, I think they're French, and it's a young couple and they're having a baby. So she's pregnant and he's filming. And this fellow, for some reason, he's very versed in technology. You know, he's a techie, and he put together this video that shows actually from the moment of fertilization at the cellular level all the way to the birth of the baby. And it's all compressed in 6 minutes, all right? So this guy calls himself Evil Cat for some reason. But the video itself is just spectacular. And also, there's no narrative really. He chose acid rock music, which is about the worst music I can think of, all right? I like classical music, I'm not into acid rock, but that's the background music. So I'm gonna cut off the sound and I'm gonna do a little narrative myself of what's going on. And I'll pause the video at some point because it's really going very fast, but the event essentially begins at the cellular level with the release of the sperm. Let's see. Oh yeah, sorry, I was gonna cut off the sound. Oh, here. There's the acid rock. Okay, now, so the sperm has just been released, and what we're looking at really is a bunch of sperm in the semen that are all navigating and wiggling toward the common goal, which is to try to get the egg fertilized. So the first thing it has to do is all the sperm is going through the cervix of, from the vagina into the uterus. Okay, so at this point, we're in the uterus. And what do you see up here? These two holes are the two fallopian tubes. So this is like the ceiling, if you will, of the uterus, okay? And this is the uterus lining, the uterine lining, also known as the endometrium. And you can see that the sperm, they're all wiggling along, and there's fluid, there's mucus there. We'll talk about that mucus, especially when we talk about natural family planning, how that mucus, the cervical mucus, is both a lubricant, it's a highway, and it's nourishment for the sperm, has many, many qualities, all right? Okay, so basically, here's fallopian tubes, and one of the things that happens is that natural selection, so 100 million sperm were released, assuming that this was a potent ejaculate, and they're out to fertilize one egg. So the competition is on, literally, and it's a race to see who gets there. Right away, because there are two fallopian tubes, the first selection is going to be, well, they have to navigate the crevices of the inner lining of the uterus, the endometrium that has cristae. They have folding in and out like that. So the sperm has to navigate all that up and down crevices. There's like caverns at the microscopic level. And that's why the distance proportionally is equivalent to hundreds and hundreds of miles for, you know, to our size. If the sperm were to be the same as our size, it would be a very long distance. Anyway, the sperm is navigating all that up against the flow of the cervical mucus, and now it's going into what seems to be the right fallopian tube on the right side. That already is a selection, that's a 50% selection right there, because half of the sperm will go into the left tube, the other one to the right. They don't know where is the egg, you know, either way, but it's a split in the road, right? And so that selects out right away, on average, 50% of the, so now we're down to only 50 million instead of the 100 million. And it's very typical of sperm because the sperm come from men, and men, we know we don't ask directions, okay? And so half of them just got lost into the wrong tube. Okay, so now it's going through the fallopian tubes. At some point, you're gonna start seeing the fimbriae, the little finger-like structures there. These are finger-like, hair-like cells, similar to the ones that are inside the intestines that do the absorption and the digestion, okay? And they have this undulating motion, undulating fashion that, again, are constantly moving toward the the uterus in a pattern is called peristalsis. It's a wave-like motion that goes through the tubes, moving downward. So bottom line is that the sperm are swimming against that motion, all right? And that's why the vigor of the sperm, the capacitation of the tail, which actually finishes the capacitation, is finalized within the reproductive tract itself as it interacts with the cervical mucus. So you'll see this hair-like structures waving this way and the sperm in the opposite direction. All right. You see? All right, so fewer and fewer sperm. Now, there's the prize, there's the egg. But the egg is not by itself, actually. The egg is called a cumulus. In other words, the egg is surrounded by 2 layers of cells, not one, but 2 layers, okay? There's the zona radiata and the corona radiata and the zona pellucida. We'll get to those, I'll show you some diagrams. They're just Latin words for layers that are protecting the egg. So the egg is encased and protected with these layers of cells that are protecting it, all right? And the whole structure is the cumulus. So now they're getting close. You can see some sperm down here. These are the fastest one, the most vigorous, all right? And they're coming up to the cumulus for fertilization. And it's gonna zoom in to show the actual fertilization process. Here is the corona radiata, which is a cluster of cells on the outside that is radiating out. And so that's why it's called the corona radiata. It's a crown, if you will, all around the egg. Similar to the spikes on the coronavirus. That's why they call corona, because it surrounds it. And the zona pellucida will be inside. It's like a jelly-like structure that also envelopes the egg and maintains it secure. Now it's going to show one sperm. See, several is trying to penetrate. One will actually make it through. And then a reaction. Here's the one sperm. There's another one next to it here in a microsecond distance. Now it has to go through the zona pellucida here, which is a jelly-like structure. And the sperm will release enzymes at the tip of the sperm, which is called the acrosome. There's a bag with enzymes, digestive enzymes that are released to bore through, literally make a hole through. And this is all happening in a split second, okay? It's called the acrosomal reaction. There, it just spilled the enzymes and now it bores through that hole. And then the next one got sealed out. There's a counterpart reaction that happens on the egg side that is called the cortical reaction. And the cortical reaction has to do with calcium balance through the egg membrane, and it seals the egg. It's almost instantly, okay? It's in a fraction of a second. Once the first sperm penetrates, the egg is sealed so that a second sperm doesn't get in precisely and deposit another 23 chromosomes. You follow? And it's a split second. And that cortical reaction needs to happen and be intact. Otherwise, we get an abnormal embryo. All right? All very finely regulated by regulatory proteins and pathways and mechanisms that happen time and time and time again every time there's a fertilization. Okay, so now we have a fertilized egg. He's going to get in the egg for a moment there. Went through, now it's penetrating the membrane, the egg membrane, loses the tail. The membrane of the sperm starts getting digested out by enzymes inside the cytoplasm of the egg, all right? But the important thing here is for the sperm nucleus to be released. And there, that's a sperm nucleus. It's called a pronucleus because it's haploid. And here's the female nucleus. And that's the egg nucleus. So here's the sperm nucleus and here's the egg nucleus. This is the second layer of linking, of yolking. The first layer of yolking is egg and sperm fusing. Necessary, but not sufficient. The second layer of linking or yolking is the 2 pronuclei, the male pronucleus and the female pronucleus, to restore diploidy, right? And so they fuse, the 2 nuclei also fuse there, bang, now we have a diploid nucleus. And the first cell division just happened. All right. And he's going to show in greater detail. That's the diploidy there. Now the chromosome is going to replicate and undergo the first cell division. Here's replication going on. Of course, this is a diagram, right? That's the duplication process there, replication machinery, the two sister strands, the sister chromatids there. Okay, so each one of these is a sister chromatid, right? And now these are going to line up at the equator. This is meiosis, I'm sorry, regular mitosis, right? That was a split from anaphase to telophase to the opposite poles, each individual chromatid. And now cytokinesis, the pinching of the cell in between. And this is the first cell division. Bang, that is within 24 hours, the first day. Okay, first cell division. Now the zygote has already split into two, and it's called the two-cell layer, the early embryo, two-cell layer. Here, we're still inside the fallopian tube at the distal end, close to the ovaries, heading toward the uterus where the embryo actually has to implant. That journey is going to take 5 to 7 days in the human, 5 to 7 days after fertilization for implantation. So now this embryo is going to start rolling down the tube by the peristaltic motion and the movement of the finger-like, hair-like cells and the flow of the cervical mucus. As that little embryo is beginning to move toward the uterus, okay, the cells within are also undergoing mitosis and splitting the second day, the third day, and so forth. So from 2 cells, we get 4, and from 4, 8, 16, 32, 64. You see, it's a geometric progression, so it's very fast. Every split duplicates. It's not linear. Linear will be 1, 2, 3, 4, 5. This is geometric. It's an exponential, all right? And so very fast, the embryo is accumulating cells, very fast. Okay, so that's the second stage. It's called the morula. We'll get into it. That's the 8-cell stage, then 16 and so forth. Now it's going to drop down the fallopian tube into the uterus. Oh, no, drop, boom. Okay, now you're going to see a hatching. You're going to see a hatching. This is a mammal. This is a human. How can we hatch? Only chickens and birds hatch, right? From an egg? Yes. But this, you can think of this as what we call the egg, the chicken egg, the hard white chicken egg or brown, whatever color. You know, this is kind of similar because it still has the cumulus layers around it. So the embryo, right, still has the zona pellucida and the corona radiata around it like a shell, but it's like a soft shell, like the shell of turtles or reptiles that are soft, like that, okay? And now you're gonna see this little embryo come out of that shell and discard that soft shell of tissue, discard it, leave it behind, and move forward. And that is a type of hatching. So we also hatch. It's vestigial. We have a vestigial hatching process, probably reminiscent of our reptilian ancestors, common ancestors between reptiles and birds and mammals. So watch the hatching. Here, it's coming from this end here. Here it goes. Blue. There. So that's the embryo, okay? And the shell, soft shell of tissue, coronary, sort of got left behind. And what happens to this? It gets digested. So nature is very economic. It uses everything, no waste. It just gets digested and reabsorbed by the walls of the uterus as nutrients. Okay, so now we have this blob that is the blastocyst, and this is what's going to implant 5 to 7 days after fertilization. Okay, this is called a blastocyst, several dozen cells, and that's what's going to bury through the endometrium for implantation. Here we go. Okay, now it's going to show just the gastrulation process, how the embryo starts splitting, dividing in half and advancing in development and creating more and more tissue that is specialized tissue. That's the neural groove down the center, bilateral symmetry. And the somites, which are precursors of the muscles and the spinal cord. The heart, very early on. These cells is fantastic because these are myoblasts. Myoblasts are individual cells of the heart. They have a pulse. That's why there's cardiac muscle, right? And cardiac muscle, they have their own pulse. They self-beat. And when they come together, but they're beating individually, and when they come together, they synchronize their beat into an actual heartbeat. And that happens the first couple of weeks of embryonic development, all right? So watch them as they synchronize. And they're very hard. Oh, so the heartbeat of the embryo, I guess I never said it, is about 200 beats per minute. Boom, 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 very, very, there. They synchronize, okay, into the heartbeat, very, very fast heartbeat. It's freaking out. You think that if you hear an ultrasound of a baby, of an unborn, you think it's having a heart attack. It's not. It's just very fast because all of this is happening very fast. And it needs a lot of oxygen and nutrients to go through the whole body for this to happen so fast. Of course, it doesn't happen in 6 minutes. It happens in 9 months, all right? But it's still very fast, proportionally speaking. So you see all the formation that's going on of the organs and tissues. By the time we get to 2 months, we have all of our organs in place, but in miniature. All the major organs, the brain, the heart, the kidneys, the liver, everything is in place, but in miniature. Of course, then it changes from embryo to fetus, and the fetus is going to be moving around, bumping. Sometimes they open their eyes, sometimes they close. They suck their thumbs, they suck their toes, they play around with the umbilical cord. They can see some light coming through, can hear some sound coming through the abdomen. And at some point, that little guy is getting so large and uncomfortable in there, he or she's got to come out of there. So they have periods of sleep and rest and moving around, and the mom can't stand it anymore, and she wants this kid out because it's just restless. Getting very crampy in there. And now they'll get into the fetal position. At the end, from 2, 3, the 2, 3. In the end, he says, and there's Evil Cat himself there, and there's mom about to give birth. And in 2006, there's a baby, I can't really tell, boy or girl, but it ends with the first bath. Started with 1 cell. Started with 2 cells that became 1, and then 1 trillions. Amazing, 9 months. It's the fastest time of growth in our life. We go from single cell to about 1 foot long in 9 months, in less than 1 year. And then we spend the rest of our lives growing maybe 6 more times to 6 feet, if that. Okay, so super fast, rapid growth in the embryonic development. And that's why it has to be protected within the mother's womb precisely to offer the most secure place possible for that embryonic development to occur. All right, a little bit of the diagram here. Here's the egg, and I mentioned the zona pellucida and the corona radiata. So the whole thing is the cumulus, right? The cluster. Here's the nucleus, the pronucleus, haploid, cytoplasm, and you see these little cortical granules. These are the ones that are involved in the cortical reaction in sealing the egg from inside. And they're little pockets of calcium, basically. So it's a calcium back and forth through the cell membrane that makes the egg seal. And I don't know this for sure, but just my intuition, a shell from a chicken egg is mostly calcium. In fact, bird eggs are mostly calcium, right? And it's hardened. So they probably have an excess of this, whereas we have a very little, and that's why it doesn't harden. But anyway, calcium is involved in the sealing of the egg at fertilization. The sperm, as you can see, morphologically, is a very, very different structure, even though genetically it's equivalent. Haploid nucleus, 23 chromosomes, unpaired, very little cytoplasm, Here's a little bag at the tip, the acrosome. The main body, or the base of the flagellum rather, is full of mitochondria that you can see lined up, because mitochondria produce energy. And this flagellum is going to need a lot of energy to do that traveling fast, right? How long does fertilization take in the human? It can be anywhere from probably 5 minutes, the shortest, from ejaculation to fertilization, to up to 5 days after intercourse. Okay, so there's a window of 5 days. Now, sperm normally only lasts 24 hours alive, just like the egg. The egg only lasts 24 hours alive, okay? And if there's no fertilization within 24 hours on average, the woman will discard that egg, and that will be the menstruation. Sperm by itself also only lasts 24 hours on average, but inside the female reproductive tract, meaning after ejaculation, that sperm can live up to 5 days. Because you see, it has very little cytoplasm, so it doesn't nourish itself. It cannot nourish itself, unlike the egg, but it gets nourishment from the cervical mucus. So the cervical mucus is nourishment for the sperm. It's also lubricant, and it's also a highway, which I'll talk more about when we get to natural family planning, for allowing the sperm to reach its goal. So the life of the sperm is multiplied 5 times within the reproductive tract. That's why a woman could have ovulated and could have had intercourse and not ovulate until 5 days after. She could still get pregnant from that fertilization, from that intercourse that happened 5 days before she ovulated. That's kind of the stretch, the maximum window, okay? And so that's why that 5 days have to be taken into account in natural family planning for the couple, whether they're looking for a baby or seeking to avoid getting pregnant. This is an actual micrograph. It's a photograph of an egg, as you can see here, with the cumulus, the zona pellucida, the corona radiata. That's a scale, 1/10 of a millimeter. That's why we cannot see an egg to the naked side. But a cow's egg, cattle egg, are about 10 times larger than human eggs. So one can actually see a cow egg, little tiny dot, which would be about a millimeter, all right? Single cell. But it's huge compared to the other cells in the body, and certainly huge compared to the sperm. Here is the scale of the sperm, 0.05 millimeters. So actually it's 1/20th. The sperm or the egg is about 20 times bigger than the sperm by comparison, okay? All right, the actual process of fertilization in steps. Here is the point of entry for the sperm, going through first the corona, and then the zona pellucida. Zona gets digested by the acrosome reaction, right? Enzymes, digestive enzymes that go mostly proteins, proteases, and eventually the sperm pronucleus gets released into the egg itself, into the cytoplasm, and fuses with the female pronucleus to form one diploid nucleus. That's the second layer of linking, right? That process is called syngamy. So you may see this in the literature also sometimes. Syngamy. Syngamy is when we have a diploid nucleus, when the 2 haploid nuclei finally fuse into one inside that fertilized egg, okay? That's called syngamy. And that is actually when the diploid number of chromosomes is restored into a single structure, into a single nucleus. So we can say that at this point, fertilization is complete. The actual process may take several hours of this process going on. Okay? The sealing is very fast, but the union of these 2 pronuclei may be a little slower. Mm-hmm. By the way, here's a polar body. As you can see, it's stuck in the gelatin in the zona pellucida, all right? And that's a polar body there. So syngamy is completed fertilization. This is an actual photograph, a micrograph of colorized, because these structures would be basically transparent to our eye. been colorized so you can see the egg there with many sperm. Perhaps this is the one that's fertilizing and penetrating. It's hard to tell. But you can see the actual photograph with the tail and everything else. So the diagrams are fairly accurate. Okay, let's put it in situ, in place. Here's the ovary, fallopian tube, which is kind of like a horn, it bends. In fact, this is called the horn of the tube, or the ampulla, this bend here. And that's where the egg waits for 24 hours for fertilization. All right, after ovulation, the follicle ruptures and the egg is captured by this little hand-like structure. It's called the infundibulum. Don't worry about that, it's too technical, but it's basically like a glove. So the fallopian tube is not literally attached to the ovary, but it hangs, it hovers over it. And there's a little gap there of a few millimeters sometimes. There's a gap, and so the glove is there waiting for ovulation to occur and to capture that little egg, okay? These little finger-like structures are called fimbri. Again, not to worry about that detail, but this is important, the ampulla. The ampulla is the bend or the horn of the tube. There are two, right? One on each side, two tubes. And this is called the distal end because it's far away from the uterus, whereas this other side of the tube is called the proximal end. So you got proximal and distal. Anyway, the ampulla here is where normal fertilization occurs and should occur, all right? And that's why I'm saying that the egg will position itself here mostly through that peristaltic motion and wait there for about 24 hours to see if fertilization is gonna happen. If there's no intercourse, then that egg over-ripens within 24 hours. It's like a fruit, you have the fruit, today was green and tomorrow it's rotten, okay? And so it's over-ripened and it's discarded. And that's menstruation eventually. And then a new cycle begins, and that happens on average every 4 weeks or once a month and so forth. If there is fertilization, then we have the process that we saw earlier in the film, and the little embryo begins splitting because now at that point it's mitosis until the end of life, right? Duplicating cells. That happens as the little embryo is moving down the tube into the endometrium where the embryo is going to implant. And again, that takes 5 to 7 days in the human. So therefore, my big emphasis in this is that implantation is a separate event from fertilization. Sometimes you have ideology coming into biology and trying to redefine biology by some kind of ideology. They're trying to justify an unjustifiable position and saying, well, fertilization is not complete until we have implantation. And that is unscientific, that is not supported by the science and by nature. And that is just to justify experimenting with human embryos. Because they say, well, if the human embryo is not implanted, then it's never fully human, and because fertilization has not been completed. And that's a big lie, all right? And that's part of the industry, in the in vitro industry will do that. So to use embryo experimentation. All right, so fertilization is complete, and then the embryo moves down into the uterus a week later. Let's look at some photographs. Here is the zygote. You see that the two pronuclei have not fused yet. They're about to fuse, all right? So syngamy is just beginning, has not ended. You can see the zona pellucida and the corona radiata, so the cumulus is still intact. In fact, you can see two polar bodies here, right? 1 and 2. Then the next stage is a 2-cell stage. The cell is splitting, mitosis, forever until the end of life. It still has the corona and the zona, so to give integrity to the embryo, precisely so that we don't end up with twins all the time, right? To maintain the embryo together. The zona and the corona stick around until these cells actually establish an intercellular connection and give integrity to the embryo as a whole. If the zona and the corona were not there, we'd have identical twins all the time, which may be what happens in other animals when they have multiple births, okay? Yeah, multiple siblings. Anyway, from 2, 4 cell stage, 8 cell stage, 16, 32, etc., until we get a cluster of cells, dozens of cells, and that is then changes name into blastocyst. Okay, so the first stage is the zygote. The second stage is the morula. So zygote, I told you, comes from zygon in Greek. Morula now comes from Latin, mora, means tiny berry. So mora, just like in Spanish, zarzamora is a berry, all right? Like a raspberry that has its little clusters. Okay, so that's berry, mora, berry in Latin. But because these are tiny, microscopic, then it's the diminutive. And morula is tiny, little tiny berry is what it means. Basically, it looked like that and the name stuck. The third, by the way, each cell, each individual cell is called a blastomere, all right? Too much detail, not to worry. Third stage, the blastocyst. The blastocyst is important because what happens at some point, that little ball of, that little cluster of cells is going to begin to hollow out inside. It's going to begin to hollow out like a tennis ball, for example, all right, that is hollow inside. The only difference is that inside that little cluster of cells, it's not air because there's no air there, right? It's fluid. It's mostly water. It's fluid, but it begins to hollow out. And that becomes then the third stage, which is called the blastocyst, all right? Also, sometimes you'll see embryoblast. Now, what happens? The blastocyst has 2 different types of cells. So cellular differentiation has already happened. In the morula, all the cells are identical. So much so that if we pluck one blastomere out of a morula, we can generate a new individual from that one cell. It essentially reverts back to a zygote, okay? And that's why we can get the twinning there. So all of the blastomeres in the morula are identical to each other. They're essentially clones. They all have the same DNA, genetic material, etc. And there's no cellular differentiation yet. But at the blastocyst, we begin to have 2 cell layers, 2 distinct cell layers. There's an outer cell layer that is forming the ball itself, okay? And that's called the trophoblast. And then there's an inner cell mass that develops. There's a little cluster of cells on the inside that is just called the inner cell mass. And the inner cell mass is abbreviated like ICM. So whenever you see ICM, that's a reference to the inner cell mass. Why is this important? Because these are the embryonic stem cells. These are the embryonic stem cells, okay? The ICM, the inner cell mass. Okay, functionally, what happens? The embryo proper comes from the ICM. The embryo, the next stage, was going to be the gastrula, will form from the ICM, okay? So the ICM will become the embryo proper, the actual embryo. And that's also why it's called embryoblast. What happens to the trophoblast, to this outer layer of cells, the trophoblast? Well, what's in more direct contact of the uterus? What tissue is directly in contact with the uterus tissue, which the uterus belongs to the mother, right? And this structure belongs to the baby, to the unborn. All right, so the trophoblast is in direct contact with the uterus, with the endometrium of the mother. So the trophoblast is going to become the placenta, okay? Here is just a diagram showing the penetration of the embryoblast, of the blastocyst into the uterine wall, into the endometrium. And what needs to happen is vascularization. Vascularization, establishing capillaries, right? Vascularization, establishing capillaries. Now, consider the predicament of the embryo and the mother. The mother's DNA is what percentage in the mother? The uterus belongs to the mother, right? So these uterine cells here, what kind of DNA do they have? Do they have the mother's DNA, the father's DNA, the embryo's DNA? The mother's DNA. So every cell in the uterus is 100% mother DNA, right? Okay, now we have this creature that is trying to burrow through, to penetrate and to embed itself into the uterine tissue, to the endometrium. What DNA does the embryo have? 50-50. 50% of the mother, but 50% of the genes or the DNA of the blastocyst come from the father. So these uterine cells recognize the mother DNA, but they don't recognize the father DNA. The father DNA is foreign object, and it's 50%. So the tissue, the uterus, is at a quandary. Doesn't know if accept or reject this other creature that is trying to bury itself into me. It could be a friend or it could be a foe. It could be benevolent or it could be a parasite that's trying to bury into me. So it doesn't know. The embryo has to convince the uterine tissue that it is a friend and not a foe, that it's not a parasite. And it does that again with biochemical signaling. Very sophisticated biochemical signaling, protein releases and hormones and so forth. But essentially, the embryo needs to convince the endometrium that it is not going to hurt or kill the host, which is the mother, okay? It's going to take some nutrients out of her by way of the placenta, but it's going to be done in an organized fashion whereby the host, namely the mother, can handle it, you know? And by handling it, the mother actually gets to have a child. In other words, gets to procreate, gets to generate another one, more or less like herself, to continue on the species. So here is a vascularization going on. Eventually, a network is established, and the placenta is established. Okay, the next layer, the next differentiation that occurs after the blastocyst, once the embryo implants into the uterus, is called the gastrula or gastrulation. That's when the embryo starts elongating and dividing down the middle with the neural groove and all that we saw in the film, generating bilateral symmetry, okay? And then during gastrulation, 3 germ layers are established, which are very important for embryology, these 3 germ layers, and they're just named ectoderm, mesoderm, and endoderm, depending on where they're located. The outermost layer, represented in blue here, is known as ectoderm. The innermost layer, represented in yellow, is called endoderm. And the middle layer is mesoderm, middle, meso for middle, okay? All of our organs and tissues are going to come from these 3 layers, and that's why they're called germ layers. These are called germ layers, different from the germ layers in the ovaries and testes. But these are the somatic germ layers. So all of our organs and tissues are going to come from these layers. For example, the skin is going to come from where? The skin is going to come from the ectoderm, right? The intestinal organs, the inner organs are going to come mostly from the endoderm and mesoderm. So bone and muscle and all that, also ectoderm and mesoderm combination. The interesting thing is that the skin comes from the ectoderm, which is the outermost layer, but the nervous system also comes from the ectoderm. And you can think of the nervous system anatomically, it's the innermost part of the body, right? How the brain develops and the neural cord and all that, that's actually encased inside bone because there's a pouring in that happens during gastrulation. This is the gastrula here. You can see it's elongated and bilateral symmetry, and this is known as the neural groove. And what happens is ectoderm is actually literally going to pour in like that, the tissues, and form a tube inside, the neural tube, and that process is called invagination. It's a pouring in of cells. It's very fascinating to see that in time-lapse photography, how the skin actually literally pours into this neural groove and generates a tube, and that tube is the precursor of the whole nervous system. The front part, the anterior part, will become the brain, and then the posterior part will become the neural cord. And then branching out would be the ganglia, and then all the innervation of the body. It's just fascinating to watch this in time-lapse photography. Okay, so we start with a single cell, which is a zygote, fertilization. We go through several stages, morula, blastocyst, gastrula, and keeps growing until the 8th week. I mentioned earlier, by the time we reach 8 weeks, all of the organs and tissues of the body have been formed. The major organs and tissues have been formed, and they're in place, but they're in miniature because this embryo is about 1 inch long. And so everything is in there, but in miniature. Little tiny heart, little brain, everything in place. And now what has to happen after 8 weeks is further growth in size, mostly continue to generate more and more cells to make those organs and tissues larger and larger to handle a bigger body. And so the embryo changes name technically, it's symbolic. It goes from embryo proper to fetus. And that's 8 weeks is 2 months, right? And how much is pregnancy in the human? 9 months. So the next 7 months is basically growth in size mostly, all right? And so it changes name from embryo to fetus. This is weeks here, 7, 8 weeks, 9, 10, 11, 12, etc. And then it switches to months. These are months, 4, 5, 6, 7, 8, 9 months to birth, all right? How much do we grow? How much do we grow in size? From the zygote, which is the first stage of development, right? From zygote to newborn, about 9 months later. Okay, proportionally, let's say the zygote is going to be essentially the diameter of an egg, right? Of an ovum. Remember what was the diameter of an ovum in millimeters? 0.1, right? 0.1, so 1/10 of a millimeter. What's a millimeter? A millimeter is like the thickness of your nail, right? So 1/10 of that thickness, that's the size of an egg, okay? Which is the size of the zygote. And 9 months later, we're about 1 foot long, right? So one time I was bored and I did the calculation, see how many times we grow from the zygote to the newborn baby 9 months later, and it's about 5,000 times larger. So what I'm saying is that the newborn baby, right, the foot-long baby, is about 5,000 times larger than a zygote, than a fertilized egg, in less than a year, in 1 month. So in 9 months, we have grown 5,000 times our original size. How much more do we grow after birth for the rest of our lives? If we live 60, 70, 80, 90, 100 years, how much more do we grow? Think about it. Let's say for the sake of easy arithmetic, let's say that the baby is 1 foot long, sometimes longer, etc. Okay, let's say the baby is 1 foot long. What's a tall person? 6 feet? Keep the math simple, right? So 6 times more. And we reach that maybe 20 years, 15 to 20 years later. That's when we're born. And so for the rest of our lives, we grow 6 times more. But for the first 9 months, in less than 1 year, we grew 5,000 times the original size. That's what I'm saying is very, very fast process. Even though it's 9 months, seems like a long time, but what is happening embryologically is just, again, fascinating. And everything is in place. You know, so the heart doesn't end up inside the brain and the brain doesn't end up down by the feet. Everything is in place and working. It's fantastic. It's just spectacular. That's life. And it happens in every single human and in every single giraffe and in every single hippopotamus and in every single fly. Anything that reproduces sexually that has to go through an embryonic development. And so that is life. So really the big question, all these two lectures, to come to one conclusion, to answer the big question at the beginning of life that has immense bioethical implications. When does life begin? Individual life, not just for the human, but also for the cheetah and the mandrill and the cockatoo and whatever animal, and even plants. Life begins with a zygote. Individual, new individual, life begins with a zygote. Biological fact, I may understand it or not, I may like it or not, but it's a biological fact. Now that we have this biological fact, which I've tried to explain in luxury of detail, we have to do a bioethical analysis based on that. Okay? Questions, comments? All right.
How long is it considered a zygote for?
I'm sorry,
How long is it considered a zygote for before it's considered an embryo?
Well, so actually the zygote is the first stage of embryonic development, okay? And I said that, so that basically when does first mitosis happen, right? No, 24 hours. In other words, here's fertilization. At fertilization, we have the zygote. And then as soon as the zygote splits, now we start getting the morula, right? Here, the two-cell stage. What has happened from here to here? What has happened? One mitosis, the first mitosis. When the zygote split into two, now we have two cells still inside the corona and all that, but now we have two cells. So now we've gone into the morula, which is the second stage. And the morula has 2 cells, 4 cells, 8 cells, 16, 32, at 32 more or less. When we get to the between 1 and 2 dozen, it becomes the blastocyst. Okay. So really, the zygote lasts for about 24 hours by itself. But the zygote is really the first stage of embryonic development, the very first stage.
So after that, after the first 24 hours, it's considered an embryo?
No, the zygote is the embryo. The zygote is the first. So it's all embryo, you know, but it's like a subcategory. It's like a teenager, it's a human, but it's a teenage human. A toddler is a human, but it's a toddler human. An adult human, but all human, okay? So that's the whole point, precisely the humanity. What I'm trying to do here is to point out and to rescue the humanity of the embryo from the zygote, all right? It doesn't look like us. Of course, it doesn't look like us, but it looks exactly how zygotes will look because it's just starting development. Okay, ready for another film? This guy, Zarias, is another geek, techie. And you've heard of the ultrasound. He is the one who developed the algorithm for the ultrasound together with another fellow who developed the actual hardware. So in any machinery today, you know, there's hardware and software, just like in our computers. There's a hardware that is hard, and then there's a software that is the programs that are running the stuff. And the one without the other doesn't work. You need the two together. And so these were two guys that were working for NASA doing research and development. And Zarias developed so the other fellow, I forget his name, but he got the Nobel Prize actually for inventing the ultrasound machine itself, which is a non-invasive diagnostic technique. Just fantastic. You know, before the ultrasound, if a person had some pain, what did they have to do? They have to open and see what's in there, what's causing the pain. Oh, look, it's a piece of, it's a pair of scissors that's in there or something, right? But today, they take a photograph, literally, magnetic photograph, ultrasound, noise, actually put noise through and capture that noise over here and see, oh, look at the pair of scissors in there, okay? Non-invasive technique. So it was a quantum leap advantage in modern medicine. And this guy, Zarias, is the one who developed the algorithm for that. So basically, they're imaging the innards, stuff that we cannot see with our eyes, but it's there. And so he's giving one of these TED Talks. You know TED Talks? You've heard of these TED Talks? All right. go there. He talks a little fast, okay?
Was offered a position as Associate Professor of Medicine and Chief of Scientific Visualization at Yale University in the Department of Medicine. And my job was to write many of the algorithms and code for NASA to do virtual surgery in preparation for the astronauts going into deep space flight so they could be cut in robotic pods.
Okay, so he's into the algorithms for robotics for NASA and simulations and so forth. Okay.
One of the fascinating things about what we were actually working on is that we were seeing, using new kind of scanning technologies, things that just had never been seen before. I mean, not only in disease management, but also things that allowed us to see things about the body that just made you marvel. I remember one of the first times we were looking at collagen, and your entire body, everything, hair, skin, bone, nails, everything is made of collagen. It's a kind of a rope-like structure.
It's a protein.
Twirls and swirls like this. And the only place that collagen changes its structure is in the cornea of your eye. In your
The
eye,
cornea of the eye.
It becomes a grid formation, and therefore it becomes transparent as opposed to opaque. So perfectly organized a structure, it was hard not to attribute divinity to it, because we kept on seeing this over and over and over again in different parts of the body. One of the opportunities I had was one person was working on a really interesting kind of micromagnetic resonance imaging machine with the NIH. And what we were going to do is scan a new project on the development of the fetus from conception to birth using these kinds of new technologies. So I wrote the algorithms and code, and he built the hardware, followed over, then went on to win the Nobel Prize for inventing the MRI. I got the data. And I'm going to show you a sample of that piece.
This invention is 50 times stronger than regular glue.
It's not the algorithm,
Keep
okay?
watching to find out how this glue works. It's not actually a glue. It's a liquid plastic that hardens with UV light.
I don't know why
in 4
this came on.
seconds. This invention is 50 times stronger than
Sorry.
regular glue.
It's stuck in here for some reason. Nice glue, huh?
You can skip all the way to the right, bottom right. Yeah.
Here?
Up above it. You should see skip ad.
I was looking for it, but I couldn't, maybe because I'm in full screen. Yeah, sorry about that. Okay, here we go. Okay. There's the egg with the zona and the corona. These are actual live filming, okay? It's magnetic resonance. See, first mitotic division. So it just went from zygote to morula. More splitting. It's time-lapse photography. That's the blastocyst, early embryo. That's actual film, you can see it beating. So they can select different tissues and organs depending on the frequency of the wave and just show like the circulatory system or the muscular system. But these are real live embryos. You can see it's filming, it shows the cells floating around in the embryonic, in the amniotic fluid. Look at that circulation. See the heart beating? Of course, it's time lapse, so it's very slow. That's the nervous system, skeletal system, circulatory, digestive system, all overlaid. That's actual life. You see how the cells are moving around, floating. The embryo, the fetus is also moving. We also have toe prints, not just fingerprints, we have toe prints also. Okay, now it's going to show the birthing process. This is actual live, okay, filming with ultrasound through the mother, or MRI, actually, it's magnetic resonance. And you can see that they have selected the wavelengths that only identify the baby, as you can see here, and the skeletal system of the mother. So this is the pelvis of the mother. Okay, you can see a little bit of the spinal cord in the back here, the vertebrae, the lumbar vertebrae. And you'll see at one point the baby actually pushes on the sacrum, pushes on the pelvis of the mother to get out, right? Head first. That's normal birth, live birth. Okay, watch. And they're filming this. Okay, so the baby twists the head. It's called crowning. First comes the head, and he has to twist to come out. There, see, the twist, and that's called crowning. And now he's going to, there, that's the crowning there. Now the head is out, literally, of the body, and now he'll push. There. See how he pushes from the back of the pelvis, sacrum, the sacrum. And now the ultrasound is going on at the birthing. You can see the head of the baby here, and the rest of the baby is still inside, so it's coming out. Isn't that amazing? What can be done, and this is with ultrasound, with MRI, magnetic resonance. Okay, so the rest of the film you can watch. He's going to go.
Stay on my mind with another around the corner, or could it be a difference?
Yeah. Okay, he goes on to talk more about the.
Actually, more, working on this, kept on working on this data.
Here's a sperm, for example, an egg, and they colorize it so it can be seen because normally it's transparent. But this is all done with magnetic imaging. The morula, the embryo, all the different, that's the heart, 4-chamber heart, how it's coming together. See, these are actual images, magnetic images, and they can select out whatever tissue they want to focus on. Anyway, he goes on to explain.
In every other part of the body, look at the complexity of the folding. Where is this intelligence?
It's the brain.
That fold can actually hold more information. So as you actually watch the baby's brain grow, and this is one of the things that we're doing right now. We're actually doing a longitudinal studies of actually scanning a baby's brains from the moment they're born, every 6 months until they're 6 years old. We're going to be doing actually about 250 children, watching exactly how the gyri and the sukai of the brain is fold, see how this magnificent cell actually turns into memories of the marvel that is us. And it's not just our own existence, but how does the woman's body understand to have genetic structure that not only builds her own, but that has the understanding that allows her to become
all of the organs,
immunological, cardiovascular, system that basically is a mobile system that can actually nurture this child with kind of marvel.
It's not a painting.
It's
an actual image.
Our comprehension.
Look, there's another. Look at that.
That is us. Thank you.
Okay. Anyway, I thought this was fascinating to show that embryonic development, how gradual it is, and it's all unfolding. So really at no stage do we say that we have gone from a non-human to a human. It's all one gradual development from the zygote forward until 9 months later we're born. These are some overall conclusions. I'll just finish with this. Embryonic development is a continuous development, tissues, organs, and systems from the zygote up to birth. And after birth, we continue to grow. It's not like we stop growing at birth. You leave a baby out on the sidewalk somewhere, and that baby's going to die very fast, because that baby still needs support. So baby is still underdeveloped, has to go through toddler, teenager, and eventually the adult stage. Adult, by the way, defined biologically, which is when we reach sexual maturity. In other words, when we start producing gametes. The first ovulation is maturity for the woman, and the first time we produce semen is the maturation for the men, which chronologically could be in the teen years, but at least biologically, that's considered adulthood, all right? Okay, folks, so that's what I have. Again, any questions or comments? Basically, we've covered here sexual reproduction, in other words, fertilization and embryonic development, but emphasizing the fact that during that whole development, we are human through and through. It's just a different stage of development, depending on what stage we're looking at. And just before fertilization, we have cells that are either from the mother or from the father, or from the man and the woman, but they're not actual parents yet because no fertilization has occurred. And then after fertilization, we have a new individual. And this, again, is a universal statement, meaning that it applies to any animal or plant that reproduces sexually. Also, you look up in any biology textbook of plant fertilization, and the first stage, the first structure is going to be a zygote, a plant zygote. Is universal because that's the beginning of the new individual of that plant. And you'll see any one of these diagrams will have the zygote. See if I can find one. Okay, so this is before fertilization. It's the egg and the sperm. What is the sperm? What's the equivalent of sperm in plants? I mentioned this before. The pollen, right? In fact, each pollen grain has two sperm inside. One sperm nuclei. One sperm nucleus actually makes this tube that bores down to reach the base of the ovary where the eggs are, and then the second sperm nucleus will travel down the tube and do the actual fertilization. I just wanted to find a diagram where the process has occurred already. We still have egg and sperm. Let's see, egg cell. There we go. Okay, fine. It is a double fertilization because of the nuclei, but basically, even in plants, you can see that the first stage of development of the new individual, the new plant, is a zygote. I know if that's too small, too tiny. Yeah, you can see it here. Okay, so it's basically a universal process. The first stage of embryonic development for any individual, for any species that reproduces sexually, is the zygote. And from then forward, it's mitosis until the end of life. Okay, well, thank you very much for your attention again. And we'll continue next week. We'll start getting then into the actual bioethical topics, abortion, in vitro fertilization, natural family planning, fetal surgery, and so forth. Okay.
I have a question,
Yes, Jorge.
but it's not related to the lesson. It's something
Okay.
separate.
Sure.
So I was wondering if you could tell us a little bit about the ethics of the corona vaccine, because I've heard some stuff that they may use aborted fetus parts for like testing,
Right.
and other stuff, like you have more chances of just like getting the virus and surviving than the actual vaccine. So
I see.
what's your take
Yes.
on this?
Good. Okay, let's put it because there is a question that these vaccines may have been developed by using tissue from aborted fetuses, all right, from human abortions. I think that we can address it in the context of when we talk about abortion, which will be in 1 or 2 lectures, and we can put it in that context, okay? Then we'll revisit again the topic when we get to the healthcare bioethics course at the end of the year, academic year, and then we'll get way more into it with regards to the origin of the virus and Wuhan and all that. But at least hold that thought until next class, and we'll put it in the context of, did the vaccine use aborted fetal tissue or not? And then some other aspects of the vaccine, okay? So we'll come to it.
Okay, all right, sure.
Thank you.
Thank you.
Okay, I'm gonna stop the recording if there's nothing else. Thanks again for participating, and we'll see you next time.
Bye, have a good one. Thank
Thank you.
you.
Guys, thank you.
Best.